bore deep into the ground. So we can go hundreds of meters deep into the ground, into the subsurface, and we get more water. So more and more people all around the world are relying on groundwater resources. And the remaining is surface water. Like I said, through civilization, we have always relied on surface water, and that's not much. Not all of it is usable. Some of it is usable from lakes and reservoirs. Swamp water is definitely not usable, and river water is what we have relied on for eons. So let's take a look at the numbers. How much usable fresh water is there? The total amount on the planet is 1.4 billion cubic kilometers. Out of that, less than 1%, far less than 1% is available for use. So the usable freshwater resources are 200,000 cubic kilometers for the entire globe, and that's a very, very small percentage of the total. Should have said all of this. Yeah. Just to remind you of another fundamental, and that is the hydrologic cycle. So where is the water coming and where is it going? You know that it comes down in the form of precipitation. So you have rain and snow and hail, and all these forms are what brings the water from the atmosphere down into the surface. So it will either run off. If it falls on land, it does two things. It can infiltrate into the subsurface and become groundwater. Whatever you see on the surface of the land, that is called surface runoff. So when it flows away, that surface runoff, it collects or accumulates either in rivers, streams, lakes, ponds, and it from the rivers and streams it goes to the oceans and seas. This is where the evapotranspiration is highest. So that's where it condenses into the atmosphere and you have your water vapor and the cycle continues. Okay. So this is our basic hydrologic cycle. What do we do in terms of usage? How are we utilizing water? We extract the water either from surface or groundwater bodies. That is called withdrawal. So whether it's a well or a pump put in a canal or a river or a stream, all that is water extracted, and we use the term withdrawal. Whenever we extract water, we use it. If I extract water from a water body, I use it for drinking, cooking, it gets evaporated, it goes into irrigation, whether it's gardens, fields, wherever it is. If the water is not returned, it's called consumption. So that water is consumed, there's no return back to the water body. If the water is used for cleaning, washing, these kinds of purposes, then the water that is going down the drain, your toilets, all these things, there is water that is going down the drain. This is called return. So these returns are the water that is being returned to the water body. It is poor. It's very poor in terms of quality because you have used it for cleaning and washing and uh, sewage, toilet, and all of these things. So the quality of the water has degraded completely, but the quantity is more or less intact. So that's our basic mass balance, you might say, on water. Let's compare per capita water use both at the individual level and the national scale. So at the individual level, you might say, how much am I using on a daily basis? You can do this calculation very simply on a daily basis. You can examine your own activities in a 24-hour day. How much water are you using? So I found that I'm using just around 100 liters per day, give or take maybe 10 liters on a daily basis. There's not much variation because we all do the same things on a daily basis. You need to drink water, you need to use water for cooking, you need to use the water for bathing, washing and cleaning your house, all these things. So at the individual level in India, we are a developing country. We don't have too many gadgets. We don't have too many cars and other and large gardens and all of these things. So if you have more area for gardening if you have more than one car. In my case, I do have one. So all these things will add up if you have a clothes washing machine or a dishwashing machine or so many other gadgets which use water. All these will add up. And so in a developed country like the US, you have 227 liters per capita per day. Again, this number is related to the economic level of the person who is using the water. So poor people, they survive on 40 liters. That's the bare minimum that the government recognizes. Anything below that is considered unhygienic because then they don't have water for bathing and washing for personal hygiene. So the minimum recommended water requirement is 40 liters per capita per day. Cap is capita. And a comfortable level at the individual person level is 100 and at least what calculation I have done. But when you think about other uses for, so this is at the residential level. Now there are common uses. So for example, I stay in an apartment building. The common area in the building has to be cleaned on a daily basis. Now that you might say is not an individual usage, but whatever water is being used has to be divided. That total amount is going to be divided by the number of residents in that building. So that contributes to the individual factor. Similarly, if you're in an apartment building and you have a huge garden around you, or you have a swimming pool, or so many other things that may be water utilizing activities, all of that will be divided by the number of residents of that residential complex. The same thing applies to official buildings, institutions, schools, all of that. Okay. So that is called per capita water use at the individual level. Let's take a look at the national level. Is that so? If I take these per capita numbers, multiply it by the national population, will I get my per capita national usage? The answer is no. For us, we are not into agriculture. The food on the table, the clothes you use, any gadgets you use, everything that comes to you as a consumer product is being manufactured with the use of water. So extraction of the raw material from the ground, the manufacturing processes, the transportation processes, all of this, and the irrigation. So the biggest user of water at the large scale is irrigation. And I'll show you numbers in a minute. So at the national scale, as the scale goes up from the individual level to, let's say, an institutional level, to regional level, state level, national level, as the scale goes up, the numbers also go up. So for a developed country like the US, it's close to 5,000 liters per capita per day. That's 4,845 liters per capita per day. For a country like ours, it's close to 2,000. So 1,669 liters per capita per day. Why is it so high? We have irrigation. We are an agriculture-based economy. Most of our economic activities or economic income comes from agriculture, and irrigation is one of the main uses of water. So we have 78% utilization of water. Domestic use. When I said individual use, when I was talking about my individual use, that is called domestic use. So I'm using water in the house for any number of activities: drinking, cooking, gardening, cleaning of the house. All these are domestic uses. So this comes under either municipal water supply. You can call it municipal water supply, or you can call it domestic water use. This is only 6%. The industrial uses in our country are much less. It's about 5.3%. The data is old, agreed, but it may be slightly higher now. A major user is power. So if you think about a thermal power plant which is generating electricity, they use a lot of water for cooling. So this cooling water is a huge amount of water, and that's 2.6% of the total. Then you have navigational uses, that's only 1%, ecological uses, less than 1%, And evaporation losses in the country are extremely high. They can range from very close to 1 or 2% to 30%. It depends on the season, it depends on the latitude, it depends on the elevation. So evaporation losses are huge and highly variable. So that is the basic accounting of water use in our country. So that should explain to you how scale matters. Okay, so what I am using at the individual level and multiplying by the population is not sufficient. There are common uses. So when you think about the food, and let me also say a few other things. I haven't made slides about it, but you should know it. That let's say you are designing for a city. If you're designing for a city, you have to think about street cleaning, you have to think about recreational areas and fountains and other scenic spots that the city creates for beautification of the city. Then you have those are the main common uses. Then you have wastewater treatment, water treatment, all of them. These things are going to be part of the larger scale use of water. So these are things, that's why I said, when you think about individual use and when you scale up, the numbers are not simply a multiplication factor. You have to add to those multipliers. Okay, and we're not going to go through that because we don't have time. We have only 4 hours for this topic, 4 to 6 hours. So we'll see what we can do, but we're not going to go that far. I've already mentioned to you that when we utilize water, so let's say we take the example of a river, we withdraw water, some of it is consumed, some of it is returned. Whatever is returned is degraded in terms of quality. Some of the quantity may be less, obviously there is less quantity because some of it has been consumed, but the quality is definitely poorer. This is what we call wastewater discharge. So for a municipality, for a city, town, whatever it is, the wastewater discharges are considered to be more than 80% of the total water usage. So this is a general thumb rule that we use in design. So let us say I'm designing for a campus, for a small town, large city, whatever it is. How do I decide how much water is required? So normally what we do is we take the minimum requirement. So let's say the minimum requirement at the city level is 270 liters per capita per day. So the municipality requirement, remember it's not individual, it has been scaled up to the city level. So 270 liters per capita per day is a good design factor. You can go a little lower. I think 130 is considered acceptable, but that is for poorer communities. That's the bare minimum. So any number higher than that is good. Anything lower than that is not acceptable. Whatever you do in terms of population multiplied by the total amount of water used, the assumption that people have been using for a very long time is that greater than 80% will end up as wastewater. We actually know that the number is much higher. So for large cities, that number can be as high as 90%, it can be even higher. Okay? So the amount of wastewater generated is very, very high. And the greater the amount of apartment complexes, and there are so many other factors. Again, too much detail, so I'll get bogged down in it if I continue. But we'll just keep in mind that 80% is the minimum again. You can go higher, not lower. Then we come to another issue, and that is off-stream use versus in-stream use. So if I am withdrawing water, that is off-stream. I'm extracting it, moving it to another location, so that is off-stream. If I have uses that are related to the use of water in its location, I don't have to remove it. That is in-stream usage. So off-stream usage involves the withdrawal or diversion of water from either surface or groundwater. You can use it for domestic municipal purposes, residential purposes. Some of them are consumptive and others are non-consumptive. Similarly, you can use it for industrial processes. Same thing, it can be consumptive or non-consumptive. What about agriculture? If I am putting water in the fields, is there any return? There is very little return of water. Unless they are flooding the fields, then you might get some return. But generally, agricultural uses are considered fully consumptive use because you may lose water by evaporation and it's being consumed by the plants. So it's fully consumptive. Energy development, like I said, the thermal power plants or any other power plants, they need cooling water. And cooling water is a non-consumptive use because it's used just for cooling and the water is returned to the surface water body or to the groundwater even. There are consumptive uses, and those consumptive uses are those domestic housekeeping uses. So they have offices and toilets and other facilities for their employees and all that. So you will have these residential type domestic uses, which happen in all offices, institutions, schools, all of that. So that will be a combination of consumptive and non-consumptive uses. Then we come to in-stream water uses. There is no withdrawal of water from the surface water bodies. What are the non-consumptive uses? One is habitat for other species. So I want to preserve certain ecosystems. So most of these ecosystems can be, let me rephrase, you can have aquatic ecosystems. They can be related to rivers, lakes, ponds, and so on. You want to maintain the water quality. You want to improve the water quality so that the ecosystem thrives. So that would be a non-consumptive water use. These ecosystems are also points of recreation. So we often go to eco parks, we go to conservation areas, conservation parks. The national parks are all part of that. So these have become recreational areas for people to go and enjoy nature in its undisturbed form. So these are non-consumptive water uses. Without this water and good quality water, you won't be able to preserve these ecosystems. Then we have navigation. You know that if the river runs dry, the boats will have no place to go. So that's another simple one. And then you have fish propagation. Many areas of the country and the world rely on uh, pisciculture, which is fish propagation. So you have the use of these aquacultures. You can have not just fish, you can have shellfish cultures as well. And then you have both for food as well as for commercial purposes. So you have oyster and pearl production and so on. So there are aquaculture, which is a broad term that includes the propagation of fish for food and other commercial uses. That is all aquaculture. So I might use the word aquaculture here. And then, like I said, hydroelectric power production. That's another in-stream use. So you can use hydroelectric power generation with the streams. So these are some examples of in-stream water uses. Let's now, so up to this point, I've been talking about water quality. Maybe we can, before I start the next point, I'm sorry, quantity. I've been talking about water quantity, and from this point onwards, I'm going to talk about water quality. Let me take a minute's break and ask you if you have any questions up to this point. Am I audible?
Yes, ma'am.
Ma'am.
Okay.
Yes, ma'am.
So please ask at least a couple of questions so that I know that I'm getting through here. Anything from any of you?
No, ma'am.
Okay. All right. So then I will continue. All right. So I have. So we're going to start with major water quality parameters. Like I said, when we think about water, we need both. We need adequate quantity and high quality. We don't always need high quality. So, for example, in ecosystems, in navigation, quality of the water is not that important. But when it comes to drinking, when it comes to industrial use, for example, pharmaceutical industries, they need water that is of higher quality than drinking water because of the nature of the process. After all, they are manufacturing medicines and so on. So, depending on the usage will determine the level of water quality required. There are certain parameters that we use um, very frequently. So there are certain basic parameters that we are going to cover over here. And there are other parameters that are specific to the water usage. I'm not going to go into those specific parameters. I'm just going to restrict myself to some of the most common water quality parameters. So this is a list, and like I said, I'm not going to go through too many of them, but we can always talk about it later. The first one is coliforms. What are coliforms? Coliforms are a type of bacteria. We use, excuse me, um, let me kind of put a couple of questions to you. I know you're not going to answer any questions, but just for the purpose of understanding, when I say a water is good quality versus not good quality, when does this issue come to your mind first? My answer would be that it comes to mind first when we talk about drinking water. Let's say I go to a rural area, I need to find drinking water. And the first thing in my mind is this water safe for drinking? Because I know it's not treated water. In a rural area, most of the time you will not find treated drinking water. So how do I know whether it's safe or not? It's not easy to answer that question. The first thing we do from a layman's perspective, we look at the water. Is it turbid? Does it look muddy? If it looks turbid or muddy, I don't want to use it. I want clear, crystal clear water, right? So that is a normal human basic instinct that we want clear looking water. And that's a very good way of deciding whether a water is good or not. After all, we've had tens of thousands of years of experience with that, right? So if it's clear, more or less, it should be safe. What has happened in modern, in the last, let's say, 100 years, we are now manufacturing synthetic chemicals. Now, these synthetic chemicals are not visible to the eye, but they can be present in the water and they can contribute to toxicity in the water. But like I said, in the rural area, as long as you have clear water and you're not drinking it for a long period of time, you're more or less safe. However, in, let's say, city conditions or even in the campus conditions, how do I know whether the water is safe or not? The first thing that comes to mind is, let's say I go out and have meals somewhere at some street vendor or something. I get a stomach upset. How do I know that I either had bad food or I had bad water? That's the normal logic, right? I have a stomach upset. Most probably, I had either bad food or bad water in the last 24 hours. That is what we need to understand. We need to understand whether there are any pathogens either in the food or the water that we are taking in. So restricting ourselves to water, we use coliforms. Now what are coliforms? They are bacteria. These are bacteria that themselves are not pathogenic. However, if they are present in water, we call them indicators of the possibility of fecal contamination. Let me elaborate on that. So these coliform bacteria are normal inhabitants of the gastrointestinal tract of warm-blooded animals. So birds are included in that. So birds, animals, human beings, all of them have coliforms in their gastrointestinal tract. And we have a technical method for monitoring them. So if I bring the water sample to the lab and I want to determine the number of coliforms in the water sample, I can do that. And there are several methods for doing that. I'll come to that in a little bit. So we define them as rod-shaped, gram-negative, non-spore forming, motile. Motile means moving, moving or non-moving bacteria which can ferment lactose. Lactose is milk sugar and produce acid and gas when they are incubated at 35 to 37 degree centigrade. If you look at the temperature, you know that this is very close to the human body temperature. The normal human body temperature is 37 degree centigrade. And like I said, they are normal inhabitants. So by themselves, they do not cause diseases unless there is a strain that has become virulent or pathogenic, which may be a disease-causing strain. The best example is E. coli. Now, if I detect coliforms in water, it means two things. There are two tests. One is total coliforms, the other is fecal coliforms. Fecal coliforms are a subset of total coliform. The total coliforms can be present in soil and they can be present in the feces of all these warm-blooded animals. So if my sample is positive for total coliforms, the possibility exists that there is fecal contamination. If I test further for fecal coliforms and I find it positive, that means for sure there is fecal contamination. Now, if there is fecal contamination, that means the possibility of pathogens. Pathogens normally, so let's say I have a stomach upset, it may be diarrhea, it may be gastroenteritis, it may be any of the common bacteria that are found around us, cholera, typhoid, all of these things are related to, they will be present in the feces. So if I find fecal coliforms in the water sample, the possibility exists that pathogenic species which cause cholera, typhoid, jaundice, hepatitis, those kinds of things, all those can possibly exist as well. So it's an indicator. That's why we call it an indicator organism. By themselves, they are non-pathogenic, they are easy to monitor. People have been monitoring coliforms for more than 100 years. And so everyone is very comfortable all around the globe. There are labs that we have methods and so on for measuring these coliforms. Okay. And these are indicator, like I said, they are very important indicator organisms for determining whether a water sample is contaminated by fecal contamination or not.
Ma'am, what is the meaning of gram negative?
Gram negative is outside the scope of what we're doing here. It's based on a gram stain. It's a staining procedure that's used for categorizing bacteria into two groups, gram negative and gram positive. So you get pink colored and purple colored cells, and there are several issues associated with gram negative and gram positive bacteria. It's a way of classifying bacteria. Okay, so let's not go too far. It's a very involved process. So it's kind of a complicated method that we use in the lab. Okay, and we're not going to go that far.
Okay, ma'am.
Yeah. So there is, what was I saying? The point for you to remember in terms of pollution is that we don't want fecal contamination of the drinking water supply, right? And the best example of that, of rather the absence of that, is the monsoon season. So in the monsoon season, very often we don't go to street vendors. We find that water logging is happening in certain parts of the city. Why is that? Why is that a problem mainly during the monsoon? That is because when you have water logging, any leakage in the wastewater drains and pipes is going to spill over into the waterlogged portions. And if the water supply lines are not 100% intact, if there are leaks or corrosion or damage to these pipes, then you get contamination by the wastewater. And that fecal contamination of the water supply can cause diseases. So it's very important not to have water logging. and not to have close connection between the water supply lines and the wastewater drains. So these are some of the things that are more important from, I would say, from the perspective that you have, that you should be aware of these things, because in the monsoon season, these things are very common. And the street vendors who are supplying food and water, they may or may not be using safe water supplies. So these are issues that most of you may have heard about, but it's worth remembering them over and over again. Then we come to how do we detect and enumerate them. Like I said, there are several methods that have been in use. The most probable number is the most common and most popular method. It's a statistical, and it's both a lab test and it's based on statistics as well. So you use fermentative bacterial test tubes. Based on the gas formation, you can determine whether the sample is positive for coliforms or not. And then you have statistical tables which will convert it to number of colony forming units or cells in that case. Yeah, probable. It's just called most probable number, which is cells per ml. Then what we prefer to use in our lab is plate counts. So there are examples over here. So I'm just going to show you these examples. These are plate counts. So these plate counts are where you have nutrient agar, we add the water sample to it, we incubate it for at least 24 hours, maybe 42 or even longer, 42, 48 hours or longer. And when you get these colonies are representative of a single cell which is multiplying during the incubation period. So these plate counts are converted to colony forming units, and each colony is considered to represent a single starting cell. So the number of cells that were present in the original sample is measured in terms of colony forming units per ml, or yes, just per ml. There are different methods of doing this. This also has been in use for a very long time. And what is even better in more recent times is membrane filtration. So let's say I want to measure the number of coliforms or any other species in my water sample. I pass it through a membrane. This is a membrane which has a printed grid on it. This membrane is used for passing any amount of sample. I can pass 10 ml, 100 ml, 1 liter, 10 liters, there's no limit. So I can pass a variable amount of volume of water sample through the membrane. I incubate it and I get the same kind of colonies. In this case, a color indicator has been added. So you get colorful colonies, which makes it easier to count. And this will tell me the number of cells that I started out with. So these are some of the methods that we use, either with plating means plate counts, or with microscopes. And the microscopes can be, so this is SEM, scanning electron microscopes. We have them, and we can use them for counting the number of cells. This is optical microscopy. You have all, I think you are all familiar with optical microscopes. You can use fluorescent dyes and get this kind of image, or you can use non-fluorescent dyes. And these are examples of gram staining. So I was talking about pink and purple cells. So you can see a mix of pink and purple over here. Some are light pink and some are dark purple. So these are gram positive and gram negative cells. And here you have gram positive cells. You can see they're all extremely dark, almost black in color. They are actually purple because of the dye that has been used. So crystal violet has been used and it's made purple. And then you have the absence of crystal violet, which makes it, then another counter stain is used, and that's saffronin. And so saffronin gives it a pinkish orangish color. So that you can see all of that in these slides. These are optical microscopic images. So that's all about the first and most important indicator of contamination. Then we come to the next indicator, and that's oxygen demanding waste. Now, sometimes you go to a river or a lake and you find that it stinks. What is the first thing that comes to mind? It is stinking. Why is it stinking? So, the thing that causes it to stink is that there is organic matter in that water which is being biodegraded by microorganisms. And in the process of biodegradation, what is happening is that gases like ammonia and hydrogen sulfide are being released. And these gases are contributing to the stench. Now, you will also find, and this is again not easy to see visually, if you have an oxygen meter with you, we have portable dissolved oxygen meters. If you had the ability to measure the dissolved oxygen levels in this kind of stinky water, then you will find that the oxygen level may be either zero or barely above zero. So the health or the cleanliness of a water is completely related to the dissolved oxygen level. Um, I, I used to use the example long time back. I haven't used it in a long time. Why do you prefer to drink refrigerated water? You may have noticed that refrigerated water tastes better than water that is warm or at room temperature. And the difference between the two water samples is simply temperature. What makes temperature make the taste better? One answer is dissolved oxygen. So as the temperature decreases, the saturation level of oxygen from the atmosphere into the water increases. And it's possible that the oxygen level being higher in refrigerated water makes it taste good. So when you think about, oh, good quality, clean water, so cold, clean water tastes better. We know that. So coming to the importance of dissolved oxygen levels in aquatic systems. We are not the only organisms on the planet that like cold, clean water. Many other organisms in aquatic systems also like cold, clean water. So that is how it goes. And the saturation level of dissolved oxygen can vary depending on temperature, depending on salinity. It can vary from 5.1 milligrams per liter to 14.6 milligrams per liter at 0 degrees centigrade and 0 salinity. And if you have organic matter in this mix, then the oxygen level will go down further. So like I said, if temperature goes down or the salinity goes down, the DO saturation level increases. And you may have heard that the minimum requirement for any ecosystem is around 4 to 5 milligrams per liter. And in cold fresh waters, especially in the temperate and Arctic regions, the requirement for oxygen is much higher. It's 8 milligrams per liter because the species that live in these cold environments have a higher requirement for dissolved oxygen. So in our case, we are tropical and temperate. Our country is located in the tropics as well as the temperate region. And in the higher latitudes, you will find that the recommended levels for the aquatic ecosystems can be as high as 8 milligrams per liter. Then we come to how do we measure the oxygen demand of any waste that is going into these aquatic systems. So aquatic bodies, aquatic systems, whatever it is. Okay. Now, the first thing we'll start with is theory, and then we'll come to some practical examples. Let us say I have a wastewater and I know exactly what the chemical composition of that wastewater is. Just so let us say I have wastewater maybe from a sugar processing plant. Then I know that the bulk of the waste in the wastewater is glucose. Glucose has a very specific chemical formula. I know the formula for glucose. I know exactly how to write that equation, right? So here is our glucose. So we have glucose. We know how much oxygen is required to convert it completely to CO2 and water. So this is glucose oxidation. Now this is complete oxidation because the organic compound has been converted to CO2. I have several other examples here. I have hexane, I have benzene, I have cyclohexane. You can take any compound. Okay. If I know the chemical formula of whatever is present in my wastewater, I can write a chemical reaction and convert it complete. So complete oxidation is possible. How much oxygen is required for this complete oxygen is what we call the theoretical oxygen demand. And that is basically how much oxygen in the water will be consumed in this complete oxidation. Now, if I'm dealing with municipal wastewater does not have one single compound. It has thousands of compounds. There are carbohydrates, proteins, fats, nucleic acids, any number of compounds, natural organic matter. So it's almost impossible to be sure that the chemical formula is known. So what we do in the lab, which is very expensive and cumbersome, is we use elemental analysis. So I need to know how much carbon, how much hydrogen, oxygen, nitrogen, phosphorus, sulfur, each one of these elements which comprises whatever is present in the wastewater, it can be analyzed. We have the analytical methods and the equipment for doing what is called a complete elemental characterization. So I can do that. Sorry. So once I know the complete proportion of carbon, hydrogen, oxygen, nitrogen, etc., then I can come up with an empirical formula. So this empirical formula can be plugged into a chemical reaction, just like this reaction. I can come up with a similar stoichiometric reaction. That will give me the THOD for the wastewater. So that can be done. And I've given you examples over here. You can do them for practice. I would expect that from you. And maybe in the first assignment, or rather in the assignment for this topic, I will ask you to do it for simple compounds like the ones shown over here. And I will also ask you to do it for mixtures. Now, that is a theoretical method. But like I said, for real world situations, this is what is required, complete elemental characterization. This is a very expensive way of doing things. And generally people have neither. In our campus, we do have everything, but for the world outside this campus, it's very difficult. It's not easy to do. So in that case, we have simpler methods. So for simpler methods, we have lab methods, which are approximations of the theoretical method. Now, this theoretical method is the maximum possible requirement, okay? Now, I want to do something easy, fast and easy, right? That's what we want in real world practice. So I want to take my wastewater sample, I'll put it at high temperature and low pH. What is the benefit of putting it at high temperature and low pH? It will be completely oxidized under those lab conditions. So we have a method called the chemical oxygen demand. We use strong oxidizing agent. This is one of the strongest oxidizing agents available, which is potassium dichromate. You add it, boil the mixture, and decrease the pH. The pH is lowered by adding acid, and it takes a few hours to complete. You need some specific equipment, so we have reflux digesters and so on for doing the chemical oxygen demand test, but this is equipment that costs a few thousand rupees, while the equipment that is required for doing a complete elemental characterization costs close to 20 to 30 lakhs. So that's the difference in cost, and that's why people prefer and have been using COD as an approximation of THOD. So it's much easier to do COD analysis rather than THOD analysis. And like I said, it's been going on for 100, at least 100 years maybe. and it takes a few hours. So within 3 to 5 hours, you have a result. So this is the method of choice for a quick and easy result. Now, I have another requirement. If I am discharging municipal wastewater into a river or stream, I want to know how the microorganisms in that aquatic system are going to utilize this organic matter and how long it will take them to deplete the oxygen, okay, in the water. So I want to know how long it takes for the oxidation of the organic matter and how much oxygen will be consumed by these microorganisms. To do a complete test for water takes minimum 1 month. 20 to 30 days is a minimum. No one has that much time, money, effort, all that, because time is money and labor. No one has that much patience. No one wants to do it. So what has been done for a few decades or close to again 100 years, I think it's been around for 100 years, we have standard conditions. Those standard conditions are simulated, these natural conditions are simulated in the lab by standardizing them. It's done for a period of 3 to 5 days. Aerobic conditions are maintained. I'll go into more details over here. And the whole idea is to simulate natural conditions to the greatest extent in the shortest possible time. I've mentioned an approximation. Here, THOD is greater than COD is greater than DOD. Remember, microorganisms cannot do what COD does because these are extreme conditions. So these extreme conditions of oxidation are not what the microorganisms are capable of doing. So that this DOD, let's take glucose as an example. For glucose, THOD will be equal, more or less equal to COD and more or less equal to DOD. It's sugar, it's highly biodegradable, so it will be completely biodegraded. And when you do a COD test with it, you'll get the same result. And when you write it down and take the stoichiometry of it, you're more or less likely to get the same result. So for a compound like glucose, the three parameters are likely to be the same. If you take municipal wastewater, on the other hand, it has a large number of compounds. Some of them are biodegradable, some of them are non-biodegradable. So BOD will take care of the biodegradable compounds. COD will take care of both biodegradable as well as non-biodegradable compounds. And THOD is not possible unless you do a complete elemental characterization. Go into some detail about the BOD test. What are the standard test conditions? So when you open a standard methods book, you will find these conditions. The first thing is it's done for 5 days. That's the global standard. In our country, we do it for 3 days, and that's acceptable. I'll come back to this point a little bit later. The global standard is 5 days. The standard temperature is 20 degrees centigrade. These are standard BOD bottles. So you can see they have a peculiar shape at the top. This is a standard bottle which has 300 ml capacity. You fill it with the water sample, add some dilution water which contains nutrients in it. This inverted cone structure, this inverted frustum of a cone is there for a specific reason. The lid that we use, the cap, is made out of glass. So the whole thing is made out of glass because glass is the best material where you get least interference in terms of the absorption of organic matter and many other issues. We want it to be transparent. We want it to be glass so that there is no interference. For example, if you used plastic, plastic would cause interference because of the leaching of organic compounds from plastic. So you have the glass stopper. This is the glass stopper. It fits neatly into this inverted cone. And because it's frosted, it's a frosted glass stopper, it makes what is called a complete water seal. So water sample is filled up to the brim over here. And then the glass stopper is dropped into it. So there is a small overflow of the water sample and you get a complete water seal. So no further intake of oxygen is possible through the air, through the atmosphere into the bottle. So that is all part of the design of the BOD bottles. The dilution water is essential for diluting the wastewater. The wastewater has very high organic matter concentration, and because it's high organic matter concentration, the dilution water is required. It will provide essential nutrients. And if you're dealing with an industrial wastewater sample, then you need microbial seed. So that will also come with the dilution water. If it's municipal water sample, it's going to have millions and billions of bacteria in it, so no seed is required. Seed means bacteria, okay? So seed is that's the word we use for microbial. Seed mainly means bacteria. So we want to ensure that nutrients and the bacterial population is sufficient so that bacterial growth is not limited by these factors. The glass bottle is transparent, but we want to ensure that light is kept out. So we generally incubate it in the dark. We do not want, remember in municipal wastewater, you'll have algal cells as well. Algal cells are photosynthetic organisms. We don't want them to grow inside the bottle for the simple reason they're going to contribute to oxygen. Photosynthetic organisms generate oxygen. So we don't want that. So we incubate it in the dark. The water seal I've already explained to you. It prevents re-aeration during the incubation period. How do we measure the oxygen demand that is exerted by these microbes? These microbes are going to, in the process of their growth, they are going to exert some oxygen demand. They're going to consume. So let me come back to this reaction. You can have it as a chemical reaction, or it can be mediated by bacteria. If the bacteria are mediating this reaction, then the oxygen is being utilized by the bacteria. How do I measure the BOD? So I have only one parameter that I'm going to be monitoring, and that is the dissolved oxygen level. So I will measure the dissolved oxygen level at the beginning of the test, and then I will measure the oxygen level at the end of the test. This is dissolved oxygen. So we have DO initial and DO drop. So within that 5-day period, I have to measure the change in DO concentration. So the DO drop in the sample blank has to be subtracted from the DO drop in the sample. And if I get results, let's say in the beginning, I have 8 milligrams per liter. At the end of 5 days, I get 0. I will throw that result out. And you may say, why is that so? Why do you want to throw that result out? The simple reason is that 0 may be negative. For example, if you write delta DO, that would be 8. But it may be more than 8. Supposing I had a starting point of 10 and I still get a 0 after 5 days. Okay, so it could have been 8, it could have been 10, it could be 15, 50, any number. So that 0 doesn't tell you anything. That 0 is telling you that it can be more. The drop can be more. So my minimum DO at the end of the test should be higher than 2 milligrams per liter. If it is not higher than 2 milligrams per liter, that result is useless because that DO drop, I do not have confidence in that result. The maximum can only be the DO saturation. The delta DO, the DO drop has to be more than 2 milligrams per liter. Now let's say I start with 8 milligrams per liter and at the end of 5 days I get 7 milligrams per liter. Bad result again. I do not have confidence in that result either because when I say 8, it's plus or minus 1 milligram per liter. So 8 and 7 are not very different. They are statistically more or less the same. So that again is not a good result. So these two conditions have to be met to determine the usefulness of the test results. So these are the standard test results. Now let's come to what we do in India. In India, you'll find that labs often, very often, use BOD test results and they shorten it for 3 days with a temperature that is higher. So they use 27 degrees centigrade and 3 days for reporting BOD results. The simple reason for that is that ambient temperatures in India are much higher. The standard temperature of 20 degrees centigrade, the BOD test was invented, I think it started in the UK. So for them, 20 degrees centigrade is fine. But for us, our ambient water temperature is much higher. So 27 degrees centigrade is what they are using. And because they want results more quickly, 3 days is sufficient. And one explanation that I heard was that the technician in the lab will set it up on Friday, and when he comes back on Monday, he has the result. So that is one explanation, which may or may not be true. Okay, so then there are some more details over here. This is how we determine the BOD. So the BOD 5 under standard conditions. So you have your initial DO result, you have your final DO result, the delta drop, or rather the drop in DO is what you are writing over here, DOI minus DO final. Excuse me. And that has to be divided by P. P is the dilution factor, and this is the volume of the wastewater divided by the volume of the wastewater plus the dilution water, which is 300 ml in a standard BOD bottle. So I would say to you that in a municipal wastewater sample, we normally take maybe 5 ml, 10 ml, even less, and then dilute it all the way to 300 ml. So that dilution factor, depending on how strong the wastewater strength is, that has to be accounted for. If the blank DO, this seeded dilution water can be put in another bottle, and that we call a blank. If you have organic matter, which we generally do not have organic matter in the dilution water, but if you're dealing with industrial wastewater samples, you may want to put some organic growth factor. So we use peptone or beef extract or beet extract. All these things are used, and that is organic in nature. So they may exert some organic, they may exert an oxygen demand of their own. So this is mainly for industrial wastewaters. If the blank DO is accounted for, then you have an additional factor here, BI minus BF, which is for the blank bottle, multiplied by 1 minus P, and the whole thing divided by P, which is again the same dilution factor. Now we come to some analysis based on these BOD results. Why are these BOD results so important? And it comes down to understanding the self-purification capacity of rivers and streams. It comes down to an understanding of how DO gets depleted when we discharge wastewater into the streams. Let's take a look at this basic equation. I'm using glucose to represent organic matter. Now remember one thing, glucose is very simple, it's highly biodegradable. Organic matter in municipal wastewater or in nature is neither highly biodegradable, after all it's not sugar, and it has varying degrees of biodegradability. Okay, so just keep that in mind, and glucose is a gross simplification of the whole idea. But since we need something to represent organic matter, so I'm choosing glucose only because it's simple and easy to understand. If you look at this reaction, you can monitor two things. I want to know how much oxygen is being consumed, right? That's my objective. The objective of this whole exercise is where is the oxygen going and how fast are we losing oxygen? So I can monitor oxygen or I can monitor organic matter. Organic matter is very difficult to monitor. We have TOC analyzers. We have analyzers called total organic carbon analyzers. They are again very expensive. They are in the range of 20 to 30 lakhs again. And a portable DO meter is about 10,000, 20,000 rupees. So it's much easier to monitor dissolved oxygen. That's the first thing. You can also do titrations. If you have the water sample in the lab, you do it by titration, and that's even more cheap. So that will cost a few hundred rupees. So These are the reasons why people monitor certain parameters and not other parameters. So we have organic matter, DO concentration. You can monitor either of them, but like I said, DO is easier to monitor. It takes a few hundred to few thousand rupees. When the oxygen level is going down, we know that similarly at the same time, the organic matter is also being consumed. Both these reactants are being consumed and are being converted to CO2 and water. So you have the 5-day point. So if you're monitoring and making graphs out of this, if you're, let's say, you have the ability to monitor on a daily basis, you can come up with graphs like these. So this is organic matter or DO. So the DO is the blue line, and what is shown in the graph is the organic matter. Now, organic matter remaining is what you see in the depletion curve, and you can very well say this is the organic matter. Whatever, if you subtract whatever is remaining, if you subtract that from C0, this is your C0, the concentration at time 0. Whatever is remaining at C5, if you subtract it from C0, you get a curve like the other one. So that is the organic matter that has been consumed or oxidized. So you can generate these two curves in the lab. I want to look at the rate at which biodegradation of this organic matter is happening. I know the BOD of the sample. Now the next thing I want to know is how fast is this material going to be biodegraded. So, so we have BOD remaining. This curve is an indicator of the organic matter remaining, and we express it because we are measuring DO. We are not measuring organic matter, we are measuring DO, and therefore we call it BOD remaining, which is another way of saying concentration of organic matter remaining. So I have some concentration L0 to begin with. This L0 is what we call ultimate carbonaceous BOD. So this L0 is your ultimate carbonaceous BOD. As it gets consumed, remember two things are happening at the same time, but we are monitoring only DO. So this organic matter is going down. Based on the DO levels being DO being consumed, I can say this is the BOD remaining, so that is LT. You can express LT as a first order. Based on a first order decay reaction, we assume that it's a first order. It's an assumption, okay? So we are assuming that this ultimate carbonaceous BOD is being consumed by a first order reaction. So LT is equal to L0 times e to the power minus KT. So this is LT and this is BODT. So this is BODT remaining and this is LT, I'm sorry, BOD, no. BODT remaining is LT and BODT exerted is what you have over here. Then you have BOD utilized. So I can reverse this curve. Instead of saying BOD remaining, I say BOD utilized. So I get this curve. So you have the same thing over here. This is in terms of organic matter. This is in terms of DO. So if I'm expressing the BOD utilization, then remember what we are measuring. We are measuring BOD5. BOD5 is how much of this carbonaceous organic matter has been utilized. And along with that, oxygen has also been utilized. So I can write it in this form. BODT is equal to L0 times 1 minus e to the power minus KT. So this is what it is. Now we have another thing to deal with. So L0 is my starting point in terms of organic matter, or you can put it in terms of BOD, either way. There is a reaction rate constant. Like I said, how fast is it degrading? That is your K value. The negative sign is the degradation and K is your reaction rate constant. So it's your first order reaction rate constant. Now we are doing it at 20 degrees centigrade, but in the river, your temperature is varying. It may go down to 5 degrees centigrade, it may go up to 30 or 35 degrees centigrade. I have to make a correction because I know that the reaction rate constant is varying as the temperature varies. So that correction has to be made. So this is the correction equation. So you have Kt at any temperature T, you can correct it, and there is a theta value. Theta is the correction factor, and so theta to the power T minus 20. So T is the temperature, whatever that is, minus 20, and the whole thing is multiplied by K20. So this is how we make our correction factor, and it's based on a very, very old report, which is very important. I'll show you why. And this is based on the Streeter-Phelps set of data as well as the Streeter-Phelps equation published almost 100 years ago. And the theta that we normally use for deoxygenation or degradation of organic matter is 1.047. Now we come to another part that I already mentioned in the previous slides. I said that if you want to do the complete test, it takes anywhere from 20 to 30 days. Let's say somebody is ambitious enough, or you're a student who has any amount of time and can do it, then this is what you are likely to get in the lab. So you take a wastewater sample, you measure the amount of oxygen consumed. You may do it by titration, or you may use a portable electrode-based oxygen meter, dissolved oxygen meter. In either case, what will you get? You will get a curve that looks something like this. And this is not a mistake. This is an inflection point that has a reason. So the first part is what you are measuring in your BOD5 test. Within 7 days, if you were to take it beyond 5 days and you were to take it, let's say, to 7 to 10 days, you will get an inflection point here. There are two groups of bacteria that are involved in degrading this material. So we have carbonaceous BOD, and we have, remember what I said in the previous slide, here is ultimate carbonaceous BOD. If you think about municipal wastewater, municipal wastewater has, to be very gross about it, you have urine, you have feces. The feces are more or less carbon containing materials. Urine is very high in nitrogen. Now, the carbon containing material is going to be food for the heterotrophic bacteria. So in the beginning, the fecal matter, which is high in heterotrophic bacterial concentration, as well as in terms of carbonic, in terms of organic matter, they will take over, they will dominate, and that's the first part of the curve. Because remember, you have two groups of bacteria, one that utilizes nitrogen and the other that utilizes carbon. So since the carbon supply is very high, and the population of these bacteria is also very high, they will dominate. And that's why you see carbonaceous BOD being exerted initially. There comes a point in time when the amount of carbon in the system is decreasing. That's going, it's being consumed by these bacteria. In the meantime, the nitrogen feeding bacteria are also alive, but they are slow. In comparison to the carbon utilizing, the heterotrophic bacteria, the nitrifying bacteria, which are the ones that are utilizing ammonia, converting it to nitrite and nitrate, these bacteria are autotrophic. They are not heterotrophic. They need oxygen. They are not photosynthetic, but they are autotrophic. They do not need organic carbon. So they feed on ammonia and oxygen, and they convert it to nitrite and nitrate. Okay, it's a two-step reaction. So Nitrosomonas is one particular nitrifying bacterial species that converts ammonia to nitrite, and Nitrobacter converts nitrite to nitrate. This is a very important point when you look at the health of rivers and streams. I will show you more about this little bit later. So in the beginning, it's the heterotrophs that dominate. After 7 to 10 days, you will find that the nitrifying bacteria have started showing their impact. And when they start showing an impact on the oxygen levels, remember we are working on oxygen, we are measuring oxygen. So both groups of bacteria are using the same thing, and that is oxygen. And in the beginning, the nitrifiers are being outcompeted because the heterotrophs have dominated, the food supply is higher for them. Eventually, as their food supply dwindles, ammonia remains in solution. These nitrifiers, which are slow growing bacteria, will start making their presence felt. And this is where you see a bigger bump in the curve. So what you see here is the carbonaceous demand that is related to L0, and the next increase in the amount of oxygen consumed is related to autotrophic bacteria, autotrophic nitrifying bacteria that are going to utilize ammonia and ammonium related compounds converted to nitrite and nitrate. So this is the whole story that happens in 20 to 30 days. You can't see it in 5 days. And if I want to know what is the nitrogenous oxygen demand, so the nitrogenous biochemical oxygen demand does not show up in 5 days. So if I want to know how much of that is going to contribute to oxygen depletion, then I measure another parameter, and that is called total Geldahl nitrogen. Now, this total Geldahl nitrogen is a lab-based technique. We use what is called Geldahl apparatus. The name is from the inventor of the apparatus. And this apparatus is basically going to measure organic and ammonical nitrogen. And you can use this particular stoichiometric equation, convert this number, TKN. You can do the stoichiometric calculation, and you will find that depending on how much nitrogen there is to begin with, 4.57 milligrams of oxygen is required for complete oxidation of this ammonia. Okay, so 4.57 milligrams of oxygen per milligram of nitrogen is required. So if I know TKN, this is also a few thousands of rupees. So these are all equipment and apparatus that doesn't require too much money, and they've been around for at least 50 to 100 years, somewhere in that region, and people have standardized all these parameters. Let's come to another point. I will finish at this point. I will finish this point and then we'll stop. So what is the importance of these nitrogen species in measuring the health or water quality of a river or a stream? Let's imagine something here. So here is a stream. I have a single wastewater point. Generally, when you look at cities and you take a boat down any river in this country, you will find multiple water discharge, wastewater discharge points. But just for simplicity, we are going to stick to a single point source of wastewater. So if I have a single point source, it is discharging municipal wastewater. As I said, municipal wastewater is mainly urine and feces and water, the dilution part. So it's organic nitrogen. So most of it, starting with uric acid and the remaining compounds, it's organic nitrogen. This organic nitrogen, as it comes in contact with oxygen in the water, it will be converted to ammonical nitrogen. So you have ammonical nitrogen being released from these compounds. So you have amino acids, you have uric acid, all these things are what will release ammonia into the water. So you can see as the organic nitrogen goes down, ammonia nitrogen is rising, and that's the first step. So the first step is organic nitrogen being converted to ammonia nitrogen. This ammonia nitrogen, like I said, is converted to nitrite and nitrate by autotrophic nitrifying bacteria. The first step is ammonia to nitrite. So you have this little peak. But because nitrite is being consumed as fast as it is being produced, this peak does not hang around for a long period of time. The final end point is nitrate. As long as oxygen is available, nitrate is your final end point. That is the most oxidized form of nitrogen. So this is what you expect in a simple system. If you had a single point discharge, and this is what is shown over here on the x-axis is your time, but for a flowing stream, time and distance are related. So the water has a particular velocity. If we assume that the velocity of the river water is constant, then time and distance are related, and this is the point of x is equal to 0 or t is equal to 0. Okay, so this is a simple method. Let's see what happens in reality. So we did a study, and we did this for River Hooghly, and quite a few of my students were involved in it. So we have Hooghly, which is flowing through two cities, Howrah and Kolkata. So it has Howrah on one bank and Kolkata on the other bank. So what was monitored was different nitrogen species. Ammonia, nitrite, and nitrate were monitored. We have the concentration on the y-axis and distance, which is also related to time, like I said, and you can see the curves. So the first thing is the ammonia curve, just like the single point discharge. Remember, here we have multiple discharges, both point discharges and non-point discharges. Just to tell you, a pipe discharging water is a point source. Surface runoff is a non-point source. Okay, so when you're dealing with a river flowing through two large cities like Howrah and Kolkata, this is a stretch of 21 kilometers. It's a 21-kilometer stretch of the river. And over this distance, there are tens of discharge points on both sides of the river, including non-point and non-point discharges. So the first thing you see is the ammonia peak. Naturally, that is going to be the first thing. What you also interestingly see is a high amount of nitrate. Now, at the northern end of the city, how can you have high nitrate concentration? So we assume that these high nitrate concentrations are associated with the use of fertilizer, and surface runoff bringing excess fertilizer from the fields is going into the river. So that may be one of the contributing causes here. This ammonia is being converted to nitrite. So you see this little peak in nitrite. The nitrite is being converted to nitrate. So you have first a decrease in nitrate, and then nitrate may also be complexing with various organic matter and other metals and so on. So that may contribute to precipitation of it in different forms, and that may be the decrease and then the next peak. So you can see the peak of nitrite and nitrate is at the same point, and then you have another peak of nitrate at the end. So you can see nitrite has been consumed, nitrate has formed another peak. So you have several peaks for nitrate because that is the final end point. So when you have a river and you're trying to analyze what is happening in the river stream, this is something that is very important. Now, okay, I don't know if I have enough time. Okay, I am going to stop at this point and we will continue in the next class. I have just enough time for a few questions. Let's do that. Questions, please. I can't hear you. If you are talking, I cannot hear you. Please unmute your mic.
Ma'am, you're going to give assignment like on Teams or it will be like live assignment?
No live assignment, definitely not a live assignment. It will be on Teams. So I think we're going to make MCQ type of assignments. Okay.
Okay, ma'am.
Yeah, we have to do that. It will happen very soon. Yes. So I think today is the 20th. So by next week, we will have our first test. Okay.
Okay, ma'am.
Yeah. Any other questions? No? Okay. Um, right. So I'm going to stop at this point. And like I said, we will continue this particular topic in the next class. And hopefully it will be smoother and it will not be a problem in the next class. Okay, so I'm leaving. I'm closing this meeting and I'm leaving.
Thank you, ma'am.